that would be amazing. So this is probably a reminder to some folks, and if for other folks who are new to our work, we are doing this particular aspect of Compost for Good through a grant through the Pollution Prevention Institute, which allows us to do a deep dive focus on assisting farmers. So it could be any level of community scale composting. Maybe it's that they want to improve the soil health of their fields. Maybe it's that they already compost and they want to do better. Maybe they know they want to compost, but they don't know how to get started. We just co-hosted an animal mortality workshop a few weeks ago and had incredible interest in animal mortality composting. Maybe you're a farmer and you want to start a new composting business. All of these things are ways that we've been engaging with our farmer communities in the past, and we hope to be offering this level of support for many years to come. It's incredibly rewarding. John will, when he starts speaking, talk more in detail about how some of the specific levels of assistance that we provide on the technical scale. We also, Compost for Good is a much broader entity too. We focus on really any aspect of community scale composting, and that, John, what's your phrase, from the backyard to the you
to the industrial scale? Yeah,
we're in the middle of all of that, and maybe John will take a couple seconds to talk about his really exciting work on a neighborhood scale composting system. So the levels of support we might offer, myself, I'm a grant writer, project manager. So anytime a grant pops up that I think is relevant, once you're in our database, we push that data, those grants out to groups that they're relevant for. We offer grant writing assistance from there. We also offer project management assistance. That's kind of my bucket. Katie, it tends to be our outreach education coordinator. So she develops materials. So if maybe you have something, a signing that you want to develop, she can help you with that. We can make connections for you. We have a lot of templates on education outreach materials. We offer presentations, etc. John can, when it's his turn to do the presentation, we'll talk more, but he is definitely our technical guru. John is always pushing our thought process in terms of community scale composting and what we could possibly compost. It's every time we talk to him, he's like, what about this? So he's always learning, always trying to broaden his incredibly deep depth of experience. So that's just a really high level of how the levels of support that we would offer. But I'd say the best thing is, if you're interested in learning more about how we can support you, reach out. I think most people we've worked with in the past, and the part I'll leave you with before I pass it off to John is that we are affiliated with 2 organizations at this point in time. Compost for Good is an entity of a non-defined entity. We are affiliated though with 80K Action as one of our fiscal sponsors, and we are also affiliated with the Adirondack North Country Association. So we kind of bridge between 2 powerhouse not-for-profits in the North Country. They are our motherships. With that, I will say thank you so much for joining us. Feel free to weigh in the chat, and we will have a Q&A at the end to take your questions. Thanks so much.
Thanks, folks, for joining. It seems to me like the group is so small that I am more than happy to be interrupted at any point in time. If you have questions, don't need to make this formal. Certainly don't need to put your questions in the chat. Just shout out. How about that?
Perfect.
All right, so we'll get started. Compost for Good is the 3 of us on the rock there. Katie is my daughter, and she's joining us today, I hope, with Rowan, my first granddaughter. So welcome. Compost for Good, this is our sort of vision statement. It's you could read it if you would like, but we try to be a lot of things to a lot of different people. So I want to take just a moment to focus on this image right here. And what I see in this image is a lot of biomass. What I have come to learn in the last decade or so is that what I'm seeing right here, the biomass per given square area above ground, is overshadowed, or at least vastly less than the biomass under the soil. And we know a lot about what's above ground. We pride ourselves in knowing a great deal about the plant world. We probably realistically only know about 10% of the plant world, but we know a whole lot more about the plant world than we do the underground world. So let's take a quick look underground. Great deal of biomass underground. If you were to extract it and weigh it per given meter, square meter, you would find more biomass under the ground than above the ground. That's a hard concept for me to grasp, and it may not always be the case, but in more productive soils, certainly in older growth forests, that is the case. We all probably know that plants produce sugars and other compounds through photosynthesis. You may not know that it's a very energy-intensive process. And in some cases, plants extrude or they pump 50%, up to 50%, in some cases maybe more, of those sugars into the soil. Now, given that this is a very energetic process, why on earth would a plant do that? So that's a question I want you to think about for just a minute. Emerging research. We've learned about the importance of microbes to human health. We are now learning, just beginning to learn, the importance of microbes to plant health. I want to emphasize this. There's an enormous amount that we do not know, an enormous amount that I don't know. So leave it at that. Just to give you a sense of the power of the microbial life beneath the ground. 1 gram of soil contains approximately this many bacteria and miles of fungal hyphae. Now, that's 1 gram or 1 teaspoon of soil. Another way to put that is 1 trillion billion individual organisms that we know about. DNA analysis of soil proves that there are species of things that we don't have markers for, molecular markers for. That is to say, there are a lot of unknown species beneath the ground. Let me go back to that. 1 gram of soil, also 1 teaspoon of soil, can contain tens of thousands of species of bacteria alone. Now, not tens of thousands of individuals, but tens of thousands of species. That is, to me, mind-blowing. So let's just talk quickly about the benefits of a robust soil microbiome. If you have a robust, healthy microbiome in your soil, you can reduce or eliminate pesticides. You can increase water holding capacity, including cation exchange capacity. That will, in some cases, reduce or even eliminate the need for irrigation. A good, healthy soil microbiome increases percolation rates. That's the rate at which water moves through the soil, eliminating a lot of the issues during rain events. All of these above have the potential to save farmers money. Lastly, increasing carbon sequestration through the production of long-term carbon is a result of the soil microbiome. I'll get into that a little bit later, but there is the potential now. It's sort of hit or miss in the United States. John and Megan, if you're from Canada, I don't know how Canada works, but carbon credits right now are hit or miss in this in the United States. I suspect that we'll figure that out, and farmers may be able to make money in the future if they have demonstrative evidence of long-term carbon. So that all benefits production systems for sure. And it also benefits, I should say, plant production system, but it also benefits animal production systems. It has been demonstrated that increasing soil microflora has a fairly significant, if not very significant, impact on increased grass biomass and also the quality of the grass. There are a couple of leaders, there are many people who are working in this field, but I'm going to highlight two that I want you to know about if you don't already. Elaine Ingham is the director of the Soil Food Web. She's a professor, or was a professor for many, many years in some university out west now, I forgot. But she really is a leader in this field, and especially the importance of the microbe-plant interactions. She's the one that's coined this term, the soil food web. Now, this image is a little bit blurry, it looks like, but I want to point out that there are at least 5 trophic levels, that is to say, different levels of energy transfer or different ways things feed on each other in the soil. Now, that may not mean a lot to you, but that makes for a very complex system. Not that we can't understand it, we do understand parts of it, but very complicated. And I'll simplify what Elaine, I think, is all about, and this hopefully does justice to her work. Bacteria break down organic matter and minerals. Fungi do the same. Both of these organisms, bacteria and fungi, produce glues that bind together soil particles. Those particles may be mineral or they may be organic. Then, at another trophic level, protozoa and nematodes eat both each other and the bacteria and fungi. In that process, they release nutrients in a plant available form. So the bacteria and the fungi literally breaking down mineral material and organic material. Those bacteria and fungi store a fair bit of nitrogen and other plant nutrients in their bodies, but the cycling doesn't happen until that next trophic level comes along and consumes those guys. So this is a fairly complicated slide if you've never seen it before, but I will simplify it by saying that there is a plant successional span that at one end includes annual weeds and on the other end includes old growth forest. There is a different kind of soil biome in each of these soil types that produce, each of these different environments that produce these different plants. So by and large, we want to shoot for an area in the middle where we have approximately the same amount of bacterial biomass as there is fungal biomass. Now, there's a whole lot more going on in the soil. This is a way to boil it down, though. And we found over and over again that when we look at early successional plants where they grow, they have a high bacterial count soil. When we do the same on the other end of the spectrum, we find fungally dominated soil. David Johnson is another person that I want to highlight. If you've never heard of David's work, I encourage you to do that. He and his wife have developed what's called the Johnson-Su bioreactor. Now, this may look a little janky and a little amateur, but I assure you this man has done work along with Elaine all over the world, some really incredible work with large-scale systems, medium and small-scale systems to demonstrate that if you focus on the microbial life of the soil, you can have significant impact on plant growth. So a little bit about going a little deeper into this notion of what's underneath the ground. There are a lot of different things that live in there. I'm going to pull out only a few: bacteria, fungi, protozoa, nematodes, and microarthropods. And I'll show you images of each. This is, I think, 400x magnification. And if you can see, Jen, can you see my arrow in this on the slide?
I sure can. Yep.
Okay. So the bacteria are the either round or oblong or rod-shaped specks in the background. In the foreground, there are a number of things. There are some mineral components here and other things, but let me show you what that looks like in motion. And if I can, well, okay, here's a glitch. I don't know how to make that larger. So maybe I can do this. Let's try that again. Okay, it's just a 9-second video, but if you look carefully in the background, you will see lots of things sort of moving around. Most of those things moving around in the background are bacteria. In fact, very little other than organic matter that's beginning to aggregate, bacteria, and mineral material in this slide. And that, by the way, is, we do a lot of soil microscopy work here. We analyze a lot of compost samples and a lot of farmers' soils, and we find very often highly bacterial dominant soils, and we aim to change that with the help of the farmers and their knowledge. So here's an image of fungi and fungal spores. So for example, where my arrow is here, that's a fungal spore that has germinated, creating a little bit of fungal hypha. Here's another piece of fungi that has grown, and in a, so how do I get to the next slide? There we go. Protozoa at another trophic level than the bacteria and the fungi. This is an example of a protozoa with an amoeba inside of a testate. That's not important. What's important is that in this particular sample, there are bacteria and there are fungi. If you see right here, it doesn't show up very well because of the shading, and protozoa. So that's beginning to look better. That's got bacteria, fungi, and protozoa. In this slide, we see bacteria, fungi, no protozoa that I can see right off the bat, but also 2 nematodes. And let me show you what a nematode looks like when they are swimming around. It's quite dramatic under the microscope. This happens to be a very, very slow one. But what I want to point out is inside of this nematode are lots and lots of bacteria. These guys can eat up to many thousands of bacteria in a single day. So as they consume the bacteria, they are concentrating plant nutrients in their bodies. And then when they die, they are releasing those plant nutrients. Okay, let me get to the next slide here. How do I do that? and microarthropods. So this is at least 400x magnification. So this is a tiny little mite of some sort. In the background, you can see lots of bacteria. And so this is, and also bits of organic material here. So this is another good sign, yet another trophic level with a microarthropod. And so this is a good sign of good, healthy soil. But very importantly, I want to talk about soil aggregation. This is really, really key. And this only happens when you've got really good aggregation, only happens when you've got a good, healthy mix of different kinds of soil microbes. You can have soil aggregation with just bacterially dominated soils, but they tend to be what we call microaggregates. So the bacteria and fungi produce those organic compounds that act as glues. Those glues bind organic material. Jen, you're in the waiting room again, but I'm going to admit you. It must be somebody else. And then under the right conditions, microaggregates form into larger clumps. And that is what gives soil its tilth or its structure. That's what makes soil really great, is this aggregation. So here's a slide of microaggregation just forming. So here are some fungi, some fairly small diameter fungi that are beginning to attract and glue things to them. Those things are bacteria, they are a little bit of organic material. So this is that process just beginning. This, on the other hand, is that process quite far along. There are both organic aggregates of fulvic acids, and we have humic acid aggregates, very dark colored aggregates. We've got protozoa, we've got bacteria in the background, we've got fungi coming out, and no doubt there is a nematode just off the slide. And by the way, when we do soil sampling, we are only sampling a tiny little bit of material, but otherwise we wouldn't be able to see all of this. So let me give you a sense, and I don't know quite how this is going to go. This is a video, and hopefully it makes sense to you. We're going to see a lot of different things in this video, and maybe I'll pause it at different times. Here we see aggregates formed. We see fungal hypha. We see a spore, another fungal spore, I think a third fungal spore. We see what may be a protozoa right here, or a cyst. I'll let it play on and go a little farther. More fungi, no, that's the same fungi. More aggregates. Just, let me see if I can back up. There's also some flagella that are swimming along in the background. See if I can get to there again. Well, I've lost him, so I'm going to move on. Oh, there he is, right there. Yep, going kind of fast. I don't want to bore you with this, but I want to see, here's a sharper image of the bacteria in the background that are moving about. More aggregation, another small piece of aggregate. Do we have a question? If so, feel free to shout out. And so this is how I spend a fair bit of my time, is looking under the microscope to see exactly what that soil sample or compost sample contains. Otherwise, if I just smell it or feel it or use another analysis, I don't really know what the microbiology is like. I think That's probably enough of that. Let's stop and move along. If I can figure out how to do that. There we go. So nutrient cycling, I've mentioned, but let me get back to it. Bacteria and fungi, they consume organic material and minerals, glue them together. Once consumed, the excess nitrogen is released into the surrounding soil in a plant available form. In fact, what we're learning is that the plant root literally sends out the right kind of sugars to lure the right kind of microorganism that will deliver what that plant is looking for. Now, this is anthropomorphizing things, but the plant literally is directing a lot of this process through the exudates that it emits out of its roots. Again, almost 50% of the photosynthates, or the sugars produced in a plant, are pumped right out into the soil. And so those, kind of hard to imagine, but those plant roots are sending out trails of specific sugars, and there are a lot of different sugars in the natural world, to attract the microbe that will lure it closer to its plant tip, or the root tip, so that when that microbe can absorb the sugars that the plant is giving, and then that microbe, depending upon what kind of microbe it is, will actually release some of the plant nutrients to the plant. Those plants then uptake these plant foods and they grow. Do a quick time check for myself here. Okay. So how do we establish that biology in our soils? And then as importantly, how do we keep that biology in our soil? So for the purpose of this talk, compost is a way to, the quickest way, not the only way, but the quickest way to actually transfer microbes into your soil. So if you have really good aerobic compost with really good biology, then we like to think of it more as an inoculum than just, broadcasting organic material. Now, this was a change in thinking for me when I was in plant science, when I was in research. The whole purpose of composting was to add lots and lots of organic material to the soil. Was that a bad thing? No. What I didn't realize, and what the world really didn't realize at the time, was that it doesn't necessarily take a massive amount of organic material to create better soil tilt or better soil structure. You can do the same thing with a small amount of high quality compost. So that's really the key. High quality compost equals a wide variety of microbes, and the only way to know what you've got is to test it under a microscope. So I just want to make sure that folks realize that there are a wide variety of organics out in the world that can be composted. We've created, the very last bullet there, we've created some of the best compost in this region, at least from my analysis under the microscope, with nothing more than urine, water, and pelletized wood sawdust. That may sound bizarre, but it's just yet another thing that we currently think of as a waste that is actually a resource. So there are a lot of different kind of composting systems out there, from the very simple, that's one that Katie built at North Country School, to static-based systems that you can build out of straw bales, to rotating drum systems. This is one that we designed a number of years ago that's on our website. And it is, if you're interested, you could take a look. It's free and available to the world. Sorry for the background noise there. It looks like my printer's going on. And then, of course, the industrial scale machines that turn lots and lots of material at a municipal scale. And the one that Jen mentioned at the start of our talk, the one that we're really currently focused on, is what we call the neighborhood scale composting system. This is also a schematic that we can send you if you're interested. And we've got now a number of prototypes of this out in the world. A number of organizations are now beginning to use these, and it looks like a pretty promising design. And we're all about simple and inexpensive, and it hardly gets simpler and less expensive than this, unless you go with a windrow system, but then you've got the issues of birds, of vermin, of bear, of dogs, of smells, of all those things that you don't want to deal with. So this is an example of one of those systems. This is a 3-foot diameter drum. And this is Katie using just a hand crank. We've got a drill-powered cranks, but she is aerating this material in the drum. And we've got numerous examples where the material in the drum, before it reaches more than 3 foot deep, it's up to 140, 150 degrees Fahrenheit. So how do you keep biology in the soil long term? Continuous living roots through some sort of cover crop or your regular crop. The worst thing is to have no living plant on the soil. Reduce soil disturbance. That's particularly important for the fungal communities underground. Maintain microbiodiversity. A direct correlation between what's growing above ground and what's growing below ground. So it's not helpful to say to a farmer, you need to plant 50 different crops on this acre of ground, but it can be useful within your crop to, in the alleyways, to plant a cover crop, or even intercrop, or in the row, plant some cover crops. And a lot of people have experimented with a lot of different combinations of crops and cover crops with great success. And supply food for the soil biology. Now that food is in the form of mulch, crop residue, compost, organic material. That organic material breaks down and forms the organic acids that feed the organic acids and the other parts of the organic material that feed those soil microbes. So how do you know if you've got good biology in your soil? You have to test it. You have to test it with a microscope. You can do DNA analysis, but that's crazy expensive, but it is a great way to know exactly what's there, at least in terms of the diversity of species. So composting has implications for climate change mitigation. The real star here is fungal biomass. It turns out that the Earth's soils are the second largest carbon sink for carbon. And that's primarily in the form of fungal biomass. But you can, if you manage a 100-acre farm, you can go from being a carbon emitter. You make some basic changes to your production system. You can go from being a carbon emitter to a carbon sink. Your soils can actually pull carbon out of the atmosphere. And not only that, that carbon in the right form can be sequestered in what we call recalcitrant carbon and can last in that form for several hundred years. That's a great way to store carbon. So primary takeaways here from this talk is it's great to look at nature-based solutions. We tended to have operated in a place where we think we know better than what Mother Nature does, and we've made mistakes over and over again. Those researchers, those farmers, those experimenters who are looking at nature-based solutions are finding ways that increase that biology in your soil. Composting is all about resource management rather than waste management. Of course, it's waste management to some. If you're a manager of a large municipal system, all you're interested in, or what you're primarily interested in, is reducing that organic waste into a sellable form. But we like to encourage people to think that all of these organics that can be turned into a high-value compost is a resource, not a waste. That includes all those things that you saw on a previous slide. Another major takeaway is maintain a continuous living root system on your soil. Minimize soil disturbance and feed the microbes with organic material. Let me go back there again. So if you get nothing out of this talk other than these takeaways, then I have done my job. These are the main takeaways. There are a lot of additional takeaways, and some of you can in the Q&A can give me some other takeaways that I didn't list here, but we thank you for this. You can always email us at Compost for Good. We've got, as Jen mentioned, a couple of Facebook pages if you want to join, Facebook groups, I should say, and visit our evolving and emerging website at 80kaction.org/compost. So with that, I'll stop screen sharing, and I would love to get your feedback and your questions.
That was great,
So,
John.
Meg's got her hand up. I'm not hearing these folks, Jen. Megan's got her hand up, but I don't hear her.
Yep, we hear you, Megan.
And now, Jen, I don't hear you. Is that just on me?
Oh, hold on. Yeah, you can't hear me at all, John.
I can't hear you.
I can.
Yeah. If others can hear you, then I'm going to leave and then rejoin, because I think this has happened in the past. Can those of you on the webinar, can you hear each other? Yeah, okay, so I'm going to quit and come back.
Yeah.
Megan, if you want to fire away with your question, though, and then we can.
Yeah, so I guess my first one was nematodes. You're saying how like they consume the bacteria. Can you select for specific, or have you guys looked at selecting for specific nematodes to select for specific bacteria?
That's actually one of the questions. That hey, Dooley, you came through as me too. That's really unfortunate. That was actually one of the questions I'm going to ask John. And what I can offer while we're waiting for him to rejoin is that there is a recommendation out there that you like whatever you have on your site is adapted to your site. So if you're going to create some sort of an inoculum of life, then it is encouraged if there's some way for you to do it with what you have locally available, because there may be 1,000 different factors that we don't even know or understand why the microbes that are in your particular area are specifically adapted to that area. So that's not exactly the question you're asking, but that's an interesting observation as I'm learning. John, can you hear us now?
I'm back with you, thanks.
Awesome. So Megan, I don't know if you'd be willing to re-ask your question.
It was mainly like, have you looked at isolating certain nematodes to select for specific bacteria? And like, maybe to go on to that, like, how do you go about that?
That's a great question, Megan, and I'm glad you brought that up. Many of us, when we hear the word nematode, we are like, ugh, we don't want those in our soils, because many of us are familiar with root eating nematodes. So what we know is there are a wide variety of nematodes out there, at least 5 different major groups of nematodes. We don't have the ability, I don't have the specialized skills to know necessarily which kind of nematode I'm looking at. They are determined by their mouth parts, and sometimes I can zero in on their mouth parts and I can say, this looks pretty suspicious like a bacterial feeding nematode, but there are fungal feeding nematodes. There are nematodes that feed on each other. And there are root feeding nematodes. Does that answer your question, Megan?
Yeah, so I guess you just throw a nematode in with a different food source and see which ones they pick.
Let me make this point. Hopefully I can make it cogently. Those folks who take a biological approach to farming, they don't get overly worried when they see a couple of anaerobic microbes or root feeding nematodes. What we want to see is a wide diversity. By and large, in aerated conditions, in soil conditions that aren't waterlogged and that have the ability to grow the microbiology, the aerobic microbes will outcompete the anaerobes or the facultative anaerobes every time. And so that's the short answer to that question. Does that make sense?
Yeah, both of them make sense. Thank you.
I think, in other words, we're not trying to select for anything in particular. What we're trying to do is create conditions for the good guys to proliferate. Hi, this is Rowan.
And by the way, Megan, if there are firms out there where you can buy tens of thousands of specific nematodes, and people do this. If you're trying to, if you're a soil consultant working with a large farm, and you know that farm is deficient in certain kinds of nematodes, you can ramp up the number of nematodes in the compost extract or the compost tea, and then either apply those nematodes through a liquid formulation, through a boom spray, or you can just make a good compost that goes on in smaller fields with just a manure spreader, and you can increase the type and numbers of deficient microbes that you're looking for, or that are microbes that are deficient in your soil.
One thing I just heard that was interesting, I just listened to a presentation about soil health, and I think a helpful perspective too is that most healthy soil is rich and abundant in these things. But before you undergo, if you find your soil or your compost is lacking in things, it's an important step to take to identify why you're maybe missing it in the first place. So maybe there's over tillage, maybe there's compaction, maybe there was a history of chemical applications, any number of other factors that before you take additional measures, you want to make sure you're not going to kill off whatever else is coming in that you might add into your soil.
Yeah, super important point, Jen. All fertilizers are, I can't think of a fertilizer, at least a commercial fertilizer, that's not a salt. They're all salts of some sort, and they're typically high salt index fertilizers. What that means is that they are poisonous to a lot of microbes. Poisonous might be a strong word, but they kill microbes. So whether it's the application of pesticides that kill those microbes, or high salt index fertilizers, or over tillage, or compaction layers, or whatever it is, that's exactly right. And I also want to make this point, if you have soils that aren't compacted and that will receive, and if you're deficient in microbiology, and if you establish that microbiology and then provide the right conditions, you should only have to do it once or twice. This is not a system whereby you have to keep repeatedly applying high value compost. You should not have to do that. And also, I want to point out that you shouldn't do anything that is against your own local knowledge. You know best, if you're a farmer, you know best your fields. And when we work with farmers, we always encourage them to experiment in a small area and then see what benefits or not there are. If there are no benefits to this, then it's a bunch of hooey. But there's enough evidence around the world to not only suggest but demonstrate that this is sound technology. But it's not. There are companies out there that want to sell you these specialized inoculums, and they want to sell you that every year. That shouldn't be something that you buy into.
Hi, John. Can you hear me?
Yep.
Yes.
This is Ariana and Mel from Fill and Buy Farm.
Hi, Ariana. How are you?
Good. We kind of have this lingering question regarding the principles. So we've been trying to do more cover cropping, but we find, to maintain having living roots and having more residue, but then we have to do more tilling to incorporate it when we're ready to plant our summer crops. And we're really searching for other options of killing the cover crops effectively, but without disrupting the soil too much.
Yeah, that's a great question. It's a dilemma for many farmers. And every farm is different. Every climate is different. Every geography is different. But I know that a lot of farmers use a variety of techniques, from crimping the cover crop to kill it, and then drilling right into that cover crop. That doesn't work with everything, but that's a solution that many have found. Sometimes, so it's finding the right combination of crop and cover crop, if that makes sense. If you plant winter rye, for example, grain rye as a cover crop, and you want to plant spring onions as early as you can in the spring, that's not going to work very well, because grain rye takes a lot to kill it. I mean, maybe it could if you crimp it and you get and if you have the equipment to get bulbs in the ground, if you're using bulbs instead of seeds, it could work. Or maybe you crimp it and drill right into that killed material. But and I won't speak any farther to this because I'm not an expert on it, but I'd love to hear from anyone else who has an example of a cover crop combination that works really well. Yeah, Blair, you're muted. Yeah, there you go.
Thanks. Yeah, John, appreciate the information. I was curious, when you talked about looking at the biodiversity in the soil, are you are you just looking for presence or absence, or are you trying to do any kind of quantification of numbers or biomass or something? Or do you just infer if the organism is there, then they're likely to have a sufficient number?
Yeah, beautiful question, Blair. Thank you for asking that. Right now, we're doing qualitative analyses. So we can tell you in general whether you've got an adequate number of nematodes, an adequate number of fungal hyphae. We don't have the equipment yet to do the quantitative analyses. It's not highly specialized. We just don't have it yet, and nor do I have the training yet to do that. I know that I have taken samples of compost and run it through DNA analysis to just be able to give a general sense of the number of different species of fungi, the number of different species of bacteria, and the number of different species of unknowns, because there's no molecular marker for those. We have done that as well. But at the moment, the services that Compost for Good offers are qualitative analyses only. So to give you an example, we take a drop of soil solution or compost solution. We will run it rapidly, or we will scan it rapidly 5 times to cover a fair bit of the surface area beneath the cover slide or the cover slip to look for nematodes at 40x, because they pop out pretty quickly. And so we can get a very good analysis of at least from that sample, if it's as long as it's representative of the sample that we've got in our possession, and as long as the farmer or whoever took that sample grabbed it from a representative sample of that particular soil type, then that's what we do. And there are to have and you may know this, Blair, I don't know who you are, but you may know that there are companies out there who will give you a quantitative analysis, but they're fairly pricey. And we're doing all this for grant money, with grant money.
Just a quick follow-up. Do you ever do anything with biochar in the soil, or have you seen any differences, impacts of that? Have you looked at it at all?
It's a great question. We do not. I know that Jen is interested in making biochar. Biochar is a hot topic right now. It is a great way to aid the composting process. It's got this enormous amount of surface area, as you probably know. Can raise your cation exchange capacity quite dramatically in the soil. But they're just like compost. Not all composts are the same. Same thing with biochar, at least in my experience. There are some really good biochar out there, and then there are some that aren't so good. But we Compost for Good has not gone down that path. No. Hi, John. It looks like you're muted. Can you unmute yourself? There we go.
All right.
Hey.
How do you prepare the slide? So you get the dirt, then
Yeah.
and then is there a solution? What is it that? How do you do that?
Yeah, it's a great question. It's pretty simple, John. Let's say I've got a sandwich bag sample of someone's soil or compost, doesn't really matter. I will make sure that's really mixed up. I'll grab about 10 milliliter from that bag. I'll put it in a tube, and then I will put in distilled water to dilute that material, be it compost or soil, 1 to 5. And then I'll gently shake that 10 times, so I'm not destroying a lot of the longer fungi, fungal hyphae. And then I'll let that settle for 30 seconds. And then in the liquid fraction, if we've got a floaty bit and a solid bit at the bottom, I go into the liquid fraction, and that's where the really small things will suspend for quite some time. So we let that settle for 30 seconds so that the sand settles out. Some of the silt even may settle out. The clays do not, and the microbes, by and large, do not.
Okay, so then you go, so there's 10 mils soil, 50 mils of distilled water.
Well, enough water to bring it up to the 50 mil mark to make for a 1 in 5 dilution. And then we take one drop, only one drop of that liquid fraction, put that on a microscope slide, and then with a cover slide, we will plop down on top of that, and then that's what we scan.
And so then you experiment with how far down that liquid fraction that you go with your eyedropper or pipette or whatever you're using.
Yeah, and what we found is that in that, or what is sort of standard when people do this kind of work, is that you don't want to let that that sample settle for too long. You want to grab just the liquid fraction, and you tend to get a good representative, representation of what microbiology is in there. Does that help?
Yep, no, that's good. Because I didn't know why, yeah, no, that's perfect, thanks.
And so the cover slide, you know, is a really thin piece of glass, and it sits on a microscope slide, and the space between the microscope slide and the cover slide is, I don't know, if somebody please correct me if I'm wrong, but probably 50 microns thick.
Yeah.
And so there are, you know, as you move up and down through the focal plane, generally you hit the bacteria just like that, and they're all in the same plane. But the larger microbe, fungal hypha, for example, may go in and out of that focal plane. So you have to focus up and down to really get the full picture, if that makes sense.
Yeah, that's it. That's useful. Yeah.
And by the way, it's not a matter of bending over a you know, a microscope for hours. I can hook it up to my laptop, and I see the imagery right on my laptop, and so it's fairly comfortable to do.
Yeah. Nice.
John, could you talk for, oh, I'm sorry. Could you talk for a few minutes, whatever you were about ready to say, but also just about compost tea and extracts and like the difference between the two and why one would want to go through the process of compost tea?
Yeah, happy to do that. What I was about to say is that in our analyses, we'll take still imagery like you saw in the presentation, and also videos, and we'll embed that into a report that goes to the farmer. Compost teas and extracts, there's a lot of confusion out there, I find. Are compost teas and extracts needed? Well, maybe not. If they're really expensive, whatever the input is, if it's super expensive, you want to take a good look at it, because farming shouldn't be involving expensive inputs necessarily. So compost extract is simply taking a bag to make a compost extract. You take a bag, a mesh bag, about a 200 micron mesh bag, put a certain amount of compost in that bag, and then put it in a vessel that bubbles air continuously. And the idea is that agitation of the air moving through that and around that bag of compost immersed in that water will liberate the microbes out of that bag and into a larger solution. And if you apply that material that you've extracted, you're applying a compost extract. A compost tea, you go through the same process, except you let the the bubblers sit for longer, and you will feed it different kinds of food. For example, there are, I think, somewhere in the order of 150 different kind of natural sugars in blackstrap molasses. Molasses, small bit of molasses, is something that people use as a food in a compost tea solution. So in a compost tea, you let that bubbler go for 12, maybe 24 hours. And what you're doing is you're allowing the microbes, particularly the bacteria and the fungi, to replicate in an oxygenated, aerated environment that has plenty of food. Another common food in this is humic acid, blackstrap molasses. There are a lot of things that one can put in there, but the idea is that you feed those microbes so they actually multiply. If you then take that liquid and apply that to your row or to your farm, you're applying a compost tea. Did that make sense? So the equipment is the same, but the tea is a process by which you're duplicating, you're increasing the numbers of microbes. An extract is simply you're extracting the microbes that are currently in a compost. So if I take a bag of poor quality compost and I make an extract from it, what am I putting on the soil? Probably a lot of bacteria, probably not much else. So people who are using compost extraction equipment hopefully know how to produce really high quality compost, because the whole idea is to take a small quantity of high value compost and use that as an inoculum for their soil. And there are farms where, my goodness, these compost extractors are like grain silos. They're many hundreds of gallons, and there might be 10 stacked in a row, and they're producing compost extract like crazy. Now, these are, several thousand acre farms, but it's amazing to, I haven't seen this in person, but I have certainly seen lots of imagery where people do this.
So is there a food, whatever, if you're making compost tea, is there a food that will different foods feed fungi or bacteria?
Yes.
So if you're making a compost tea, you want a food that feeds fungi, but not necessarily bacteria.
Yes, or you put a smattering of a lot of things in there, because what you're trying to grow out are tens of thousands of different species. Some of those species may not respond very well to that particular food type that you're putting in there. Others might. So a lot of this research is still in its early phases. So a lot of people will put in a wide variety of food stock into their compost bubbler, could be any size you want, and to feed as many aerobic microbes as possible. Really key to reproduce the aerobic microbes, not the anaerobic microbes. So if you let that bubbler go quiet, then fairly quickly that liquid starts producing anaerobic bacteria and other organisms. Are they necessarily bad for your particular production system? I wouldn't know. I mean, there's so many thousands of species out there that, but what we do know is that if you try to replicate aerobic microorganisms, then that tends to work. That does work best for producing soil with good structure and soil with good nutrient cycling, and soils that are more productive. I hope that made sense.
Yeah, no, that's great. Thanks.
And by no means am I the only one with any experience here. So if anyone else has questions or challenges, by all means, or more additional information, by all means, speak
I
out.
have a lot of questions.
Hi, Dooley.
Hello, John. Thanks. So there's a group called Compost Works that sells compost for teas and extracts, and they have bacterial and fungal groups. And can I trust it, or does that have to be tested to not get bad guys?
So the general answer is the world is full of scammers. And so, or people who don't know what they're doing, maybe they're not scammers. So always, we always recommend that whatever compost you buy, whether it's just for direct application or, sorry, if you buy a couple of cubic yards of compost and if you're not particularly interested in increasing a particular kind of microbe, then who cares? Apply it, and likely it's going to be good. But if you're buying a small amount of high-priced compost that's said to be of complete microbiology, then you want to make sure it's tested. Now, you don't necessarily have to test it right before you use it as an inoculum. You might, if you know the company, you might trust them well enough to know. But if you say, can you provide me a visual evidence that what you're selling me is biologically complete compost, and if they say, well, no, we can't do that, then I don't think I would trust that company. So there are a lot of companies out there that sell compost for teas and extracts. One in particular, out in California, will sell you a 1 cubic bag, and they'll mail it to you, 1 cubic foot bag for $200. Now, is that a ripoff? It really depends. If you're using that to make a compost tea, and if you know what you're doing, and if that compost is really as biologically complete as what they say it is, it might be worth your investment.
Yeah. Okay. So then, as far as how to kill a cover crop, what do you think of solarizing?
Yeah, you and I have talked about that, and I think that's a great great method. Using UV plastic that doesn't allow a light in is another potential way. Solarizing, where you're allowing the UV radiation to come in and literally heat up the soil. The thing that I worry about with solarization, Dooley, and I don't know enough to know whether it's a real concern, is that if you overheat the ground, you're going to push that thermal energy into the soil by some amount. How much, I don't know. If I were doing it, I think I would want to take a thermometer, and I would want to know what that temperature profile looks like outside of that solarized area and underneath the solarized area. If you're hitting high temperatures at just a half an inch, I'm not sure I'd worry too much about that.
Yeah.
If you're hitting high temperatures deeper than that, it all depends on soil type and moisture content and the type of, and the amount of solar insulation that you get. But that's the thing that I worry about, that and the amount of time that it takes. But whatever works for you, I think it's great. And I'm really curious to see how your experiments work this growing season.
Okay. Well, and then I guess what we're trying is to do less tillage, but I have weeds, I cannot not till, is to fork the deeper roots by hand and then do a shallow chisel plow 4-inch.
Yeah.
So I disturb the top inches, but the bottom won't. So that's sort of my way of trying to get there.
Yeah, and that's a really good point. And I want to make sure that that what you saw in the slideshow wasn't a bunch of prescribed measures that you must take. tillage is perfectly appropriate in certain situations and sometimes necessary. The point is, the less you disturb that soil, the better for your fungal communities. But you know, if you've got a way to aerate that soil and just slice off the top portion of weedy material, especially at the white root stage, then you know that's a pretty good system. If you dig any deeper, you simply liberate, especially if you've got a large weed seed bank in your soil. Every time you disturb the soil, you're pulling up thousands, if not tens of thousands, of certain kinds of weeds. As you well know, there are weeds out there. One plant will produce 10,000 seeds. Whether they're all viable or not, I don't know, but it's it's pretty amazing. The other thing, Dolly, that we've talked about is if you can get more fungi into the soil profile, if you can change your fungal to bacterial ratio, then the weed seeds tend not to grow as robustly in those environments. So weeds are really best adapted to those early successional soils. Those early successional soils are highly bacterial. And so a really great experiment for you, I think, would be to try to take a section, doesn't have to be a large section, and really incorporate some high fungal biomass, inoculum, compost, pea, extract, whatever it is, into that soil. And then at the end of the season, see whether that's made a difference. But sometimes you know, you got to do what you got to do. But we always encourage people to experiment using your knowledge, using what you're learning in webinars like this and others. Experiment and find what really works in your environment.
Julie,
I
have you
guess my-
used any winter kill cover crops ever, like that would just die with the frost?
Sometimes, yeah. Well, I have not, when I don't get a cover crop in, I have chickweed, I have shepherd's purse, I have spring weeds that are up now that are bigger than scufflehorn, you know. But what I was asking with this question was by trying to keep the lower soils undisturbed and kind of work my way to the top, if that would encourage fungus, or do they need the top inches?
In a really robust soil with really good structure, you'll find fungi down to a great depth. But the key is to get them there to begin with. It is truly remarkable in Midwestern prairie soils, for example, that haven't been disturbed very much, how far the root systems of the plants go down and how far the biology goes down. We're talking, you know, many, many meters deep. And so as long as there are plant roots down there that are providing the sugars for these things to live, then you can grow good biology at lower in your soil profile.
And Julie, we owe you a visit too. So we hopefully can be in the field with you soon. Did you have more questions, Julie? I know Norley just asked an excellent series of questions in the chat. Julie, are you good for now?
I'm good for now. That was great. Thank you.
Okay, awesome.
And it
Yeah,
looked like Sawyer, you had your hand up.
Yeah. Hello, everybody.
Hi.
I am curious, towards the beginning of the presentation, you noted like tens of billions of trillions of bacteria in the soil. And I'm curious, like what kind of what type of soil that data comes from, and like how, like, yeah, is it loamy? Is it a compost sample? How does like soil full of clay compare in that?
Yeah, this is using USDA data, and it comes from loamy soils. It's not to say that tight soils with a lot of clay can't grow good biology, or even in tight soils that there is no biology. There is, particularly bacteria. But all soil types, almost all soil types, in almost all conditions can be improved by the addition of soil biology. So one of Katie's favorite comments, if your soil is got too much clay, what do? You add compost. If your soil is too sandy, what do? You add compost. Now, that's a general rule of thumb, and getting organic matter into the soil is always good, but more important, or as important, is getting good biology in the soil. So that particular data comes from USDA, and that's generalized data. So, but there are a lot of, if you type in how many microbes are in a gram of soil, you'll get data from lots of research institutions, the federal government, and there's some differences in those numbers, but they all indicate that there are an overwhelming number of individual microbes in a teaspoon of soil. And presumably even more, well, not presumably, even more in a teaspoon of good compost.
Awesome. Norlia, do you want to unmute and ask your question? You have a couple about, I know we've talked to you about this before, the shredding of your vines, and also about testing. Oh, she said she doesn't have a microphone. Okay, I will, John, can you see her question, or I can read it to you?
Why don't you read it so everybody can hear it?
Sure. So hi, Jen, this is Norlia, who we've met with in the past for Maples Vineyard up in the Champlain area. We are almost done with pruning now, and we have a lot of cuttings. Was thinking of if we can get a chipper to shred the cuttings. Also, we would like to do a soil test. I contacted Cornell Extension, and they said they will come for the sampling, but can I send soil to you to see for the microbes, to see for.
So last part of the question first, having conventional soil test gives you some information, some good information. And so we always encourage farmers to do that. And yes, you can absolutely send us a sample of compost. Even better, if we can find the time, and if you have the time during the growing season, we would love to make another trip up there, and we can take soil samples under your supervision ourselves. In terms of your first question, I think the report, Katie, correct me if I'm wrong, but the report that we sent did list pros and cons of using a chipper for pruned grapevines, right?
Oh, sorry. I think we gave some options for that, and yeah, how that might.
Yeah, and one of the takeaways, if I remember correctly, is that standard wood chippers probably aren't worth it, because grapevines would wrap around that chipper. There are two different types of chippers primarily. Using wood along with vines might be a possibility, but the other cautionary note that I think I put in that report is that anytime you have vining materials and a wood chipper, you run the risk of having that vine wrap around someone's arm or leg and cause a problem there. The other option that I think we listed was to simply take those pick up those those prunings and place them in a pile. If there's a way to compress that pile and make it at least brush together a little better, then there is a possibility of composting all that together. But that's grapevines are a particularly challenging one for me to think about, because I don't know of any really good equipment for grapevines. I would love to, and John, you mentioned when you and I were talking about in the agricultural world, you can get machinery that will squash things together under two rollers. What are they called?
Roller mills.
Yeah, roller mills. So that's something, I hope I pronounced your name correctly. Nora Lee, is that correct, Jen and Katie? Or Nora Lai?
Nor Leah.
Nora Leah.
Nor Leah.
Hi, John. I'm on let me just close the other one. I'm on my phone.
Oh, great.
Okay.
So-
Look, we would hold on. Let me just close that because it's echo.
Yeah.
Okay. Look, we were also afraid of contaminating the vines with the chip. If I think what you say is great. Maybe what we do is compacting it down and then composting it. The problem with the chipping is also we were thinking we do not want to, if there is any kind of diseases, it might then be spread on the vines again. So we will not probably want to spread those chips or the whatever that is on in the cuttings back to the vineyard unless after probably composting it. So I at this point we don't know what going to do. See, so
Yeah, it for me, thinking through this is a little bit challenging. And I think it was Phytophthora that you were particularly, or Fusarium, which one was, it doesn't matter. You have to be concerned about those things that would damage your crop. There's no question about that. And you have to do what you think is best. It is well known that a thermophilic process will kill those pathogens, but you've got to demonstrate to yourself that that's actually true. And that compost, should you choose to mix in manures, and that was the another thing that you were reluctant to do with good reason. But in the final product, if you can get that material up to 130, 135 degrees for a sustained period of time, and if you can convince yourself that everything in there composted well, then if it tests out well, and then maybe it's worth considering using. The other thing is that I don't know how it would work in your situation, but we are prototyping these 4-foot diameter round culverts that are 4 feet tall. And I would imagine you could, and they'll hold about a ton, Katie, or 2 tons, a cubic yard of material, and you could squash those, pack those vines inside of this container that's away from your crop, so that there's no potential to spread anything from one area to another, or you minimize that potential anyway. And then add to those vines enough high nitrogen material to decompose those vines. You might find that you end up with, either through time or temperature, an end product that you're really happy with. you've tested
Okay.
it, you've had it tested, there's no pathogen in there for you to worry about, then what do you do with it? If you're still worried, maybe it's and if you make enough of it, maybe it's something you sell to an outside audience. You sell fine wines and you sell, highly specialized bags of great compost.
Compost.
Yeah, I mean,
idea.
people are making a significant amount of money. It's all about marketing, and the marketing angle, and you've done a great job up there. So I just hate to see these things go to waste.
Yeah.
If it takes too much labor, it's not worth it. If you can find a system where you've got to do something with them, and
Yeah.
what you've been doing is burning them, which is fine,
Mhm.
but it still
Mhm.
takes labor to do that.
Yeah.
If you've got
Money
a
too.
yeah, and so burning is the last solution that I would look for. Not that it's not an appropriate solution. It might be the best solution for you, but creating a product that has more value than the mineral content that's left behind after you burn them, it makes more sense to me.
Yeah. I just want to know, you were talking about this composter. Is that something that we can procure, or how do we go about that?
Yeah, if you will help me, if somebody will help me remember, either I or Katie can send you
Yeah.
the schematic that Katie drew up,
Mhm.
and it's a do-it-yourself kind of deal. We've got people we're working with who we're procuring the drums and helping them get them assembled, but it's a pretty simple process. And
Okay.
all it takes is a drum, but
Mhm.
it takes you can't buy a 4-foot section
Yeah.
of a 4-foot diameter drum. You have to buy the whole drum. What's the cost of that drum? It's $2,300 from VP Supply, which is close to you. So if you had the need for 5 of these composters, then 5 of those composters, which is, you cut that 20-foot drum into 5 pieces, then that's then you have a small, not small, you've got a fairly large composting operation there. And so you can compost wood chips in a style like David Johnson does. You can mix in your vines. You can add manure, which you've got access to, and we can help you with a recipe development. Not saying that's the best solution for you, but it might well be.
Let's come up to our place again, and we'll see what we can do. And yeah, it sounds interesting. And look, we just finished the cutting the vines, and actually there's lots of my people has already like putting it all up into the hill. That's I was thinking like, what are we going to do next? So and when summer comes, we still have to do another pruning of all the leaves, which we have to deal with. So yeah, if there is a time that you can come up, let me know.
Do you remind me, do you have a backhoe for one of your tractors?
Backhoe, yes.
So I don't know whether it would be appropriate, but there's the possibility of packing these vines down. You'd have to add something else to it in order to make them stick down, but there's a possibility that you could fill up one of these 4-foot diameter, 4-foot high drums with a lot of vines if you had a way to gently pack them down. So something to think about. But we'll send you the diagram, and you and your husband can noodle on it and see if you think it makes sense. And if you have questions, we're happy to try to answer those questions. But we've got a number of prototypes out there that are running quite successfully. Nobody's doing great cutting, so so you'd be the first. for us
Okay.
anyway.
Okay, that'll be great. That'll be great. Thank you, John.
You bet.
Well, we are about 8 minutes past the 1:30 time, but you know, time is irrelevant when you're having a great discussion. But we want to be mindful of everybody's time. So thank you all. As John shared, please reach out to us at any point in time. You know, we've got a couple of people here that we definitely will be following up with. For folks in recording land, thanks for listening all the way through. Same thing, if you ever want to reach out to us, some of you I know who are registered, we've already are engaged with you and have some follow-ups in the horizons, but others reach out with questions, setting up a meeting consultation anytime. We will be sending recording out for folks on the call, and some maybe a few resources too for consideration. Otherwise, any final words from anybody?
Well, I just
Thank
want to
you.
thank everyone for your time. Absolutely.
Awesome. Cool. Thanks, everybody. Happy growing, and we will be in touch shortly.
Thanks, everyone.
Take
Bye-bye.
care. Thank you.
Thank you. Bye-bye.
Bye.